All right, welcome back, everybody. So it's my pleasure to introduce our next speaker, Dr. Mark Pittenger. Hi. Okay. Dr. Pittenger is a cell biologist with over 30 years of research experience with extensive training in cellular and molecular biology, biochemistry, adult stem cells, and regenerative medicine. He's been involved in the biotechnology industry since 1994. Out of many things that he's done during his career, for example, he's spent over 10 years at Osiris Therapeutics, and he's served as a visiting professor at Johns Hopkins University School of Medicine. He is currently a consultant to the Department of Surgery at the University of Maryland School of Medicine, where he continues to study the effects of mesenchymal stem cells on cardiac tissue repair and improvements in cellular therapy. So please join me in welcoming Dr. Mark Pittenger.
Well, I'd like to thank the organizers and thank you, Dr. Lau, for the introduction. So I started working on mesenchymal stem cells back in '94. I was up at Cold Spring Harbor Labs and basically a cell and molecular biologist, and was interested in these cells I had never heard of before called mesenchymal stem cells. So I was interested in staying on the East Coast and moving back to Baltimore, so I took a position with Osiris Therapeutics. And early on, I met Arnold Kaplan, who essentially named the cells mesenchymal stem cells. And I also met Stan Gerson. So Stan was an oncologist, and he's the guy who pulled my bone marrow. So I might be one of the few people who have actually isolated their own mesenchymal stem cells from their own bone marrow. So it's not entirely painful, but you don't go back too often, let's just put it that way. Stan's an oncologist, and really at that time, even in '94, University Hospital and Case Western had already infused these cells into people, so we knew there was some safety with these cells, but there was no efficacy, and there were very few assays for these. So I sort of took on that role of trying to characterize these cells and come up with better assays for their characterization. As you might imagine, the FDA requires us to characterize our cell products in many of the ways that you've seen other cell products characterized, and even other drugs. And you want to know the identity, the purity, the viability, the sterility of the product, the potency of the dosage, and the stability. And these things for cellular therapeutics are a little bit problematic, a little more complicated than they might be for small molecules, but really they do fall along these same lines, and really it comes into a consensus at some point that the purity, you want to know what else is in the sample. The viability, of course, we think is important, and so we do test for that. Sterility, just like any other product, you control for that. The potency is probably the most difficult thing because you'd like your assay to reflect what the cells are being used for medically. That's not always possible, and the FDA does understand this, but they do like you to have some measure of what you think the potency of the cells will be. And of course, this does affect the dosage that you might want to give a patient. And of course, the stability is another thing that they always want to know about. So all of these require reliable assays, and these are things that we also want to do for strong science around the lab and around the hospital. So this is an example of the expansion process for the cells. After the bone marrow is isolated, we take a band off of a phycol gradient and plate it into several large flasks, and then we put it into one of these 10 stacks. And when you isolate the cells, in this case, that's about 100 million cells there. That's actually my little finger right there, just to give you an idea of the quantity of cells. And this is a typical dose in MSC therapy. So this is, the last little bit of your little finger given throughout the whole body by an IV infusion. And that's what we're looking at in terms of therapy. So the cells are very potent, and now it's a matter of finding the best way to use them. So in the world of adult stem cells, there's lots of different varieties. Hematopoietic stem cells, endothelial progenitor cells, the MSCs, neural stem cells, iPS cells, which are gaining a lot of notoriety. But among what we call the mesenchymal stem cells, these can be isolated from bone marrow, from adipose tissue, also from dental pulp. Umbilical cord blood actually has very few MSCs, but you will see some reports of that. But the umbilical cord tissue, I think, is a very good source of MSCs. And one of the reasons is that it's this neo tissue, so it's very new, and it's not going to be used for anything else. It's going to be thrown out. So that's a good choice. And also the placenta is used to isolate MSCs. Again, another large piece of tissue that's just going to go into the waste bin. Any other tissue can also isolate some population of MSCs. Historically, we've used bone marrow because we considered a reservoir of the cells which is renewable. But in this realm, microvascular parasites, which are along the vessels of all tissues, blood vessels of all tissues, are thought to be a variety of mesenchymal stem cells. So you can imagine every tissue has some of these cells present in them. And in children who heal very well, either breaks or skin abrasions or cuts, probably have a much more abundance of these microvascular parasites, variety of MSCs. So these continue to be important, and you can think of those. They're all throughout your body. They're available at the first sign of inflammation or insult to the body. But the bone marrow is probably the reservoir for the majority of these cells. I wrote down these so I would not forget to say them, but the MSCs are more rare than hematopoietic stem cells, 10 to 100 times more rare, but they grow well in culture. So you can culture them, 20 populations is possible, even more, and there's, you can easily get 100 million cells, probably in about 3 weeks from the bone marrow draw. MSC-like cells are found in all tissues as part of the microvascular parasites. They produce growth factors and cytokines and anti-inflammatories, and that may be their major near-term use for these cells. They work very well in bone repair, which I won't really talk about, but most people don't need good bone repair. That is, there's not a big market for it. The fusion of the cells results in great cell loss. So this is true with these in vitro cultured cells. Once they've educated themselves as to what it's like to live in vitro, then you take them and you throw them in vivo, and very few of those cells survive. So this, I think, is also one of the problems that we need to work on. The adipose-derived stromal vascular fraction you'll hear reported on. This may provide enough autologous cells without the need for in vitro expansion. So again, I wanted to just make you attuned to this number, which is 3 times 10 to the 13 cells. So in a typical human body, you have about that number of cells. This is a very small number of cells. It's hard to imagine how this is going to have an effect, but as you'll see, it can have dramatic effects. This is another report that, again, places this number somewhere around 3 times 10 to the 17th. And these different organs have a certain number of cells. So even if you directly infuse these cells into particular organs, it's a very small number of cells that you're infusing relative to the total number of cells there. So I think the MSCs can also be used as a resource for understanding adult stem and progenitor cells. And at this point, there's probably been 25 or 30,000 papers published on mesenchymal stem cells. So the body of knowledge that's available is quite large. And this is really what I'll talk about, that the MSCs are more homogeneous than other stem cells. We have this argument, debate, discussion in the field as to whether the mesenchymal stem cells are heterogeneous or homogeneous. I think when you think about stem cells, they're cells that are poised for change. They're cells that are always looking for signals coming in, and so that you can quickly have changes in gene expression, but that doesn't necessarily mean that they're heterogeneous. I'll go through these as we move forward. The nomenclature stems really down this lineage that the cells were originally described as colony-forming units, fibroblastic, in the 1960s by Alexander Friedenstein, who was really the first one to culture bone marrow cells that grew attached. And he grew these from guinea pig and later rabbits. But Arnold Kaplan was the first one to isolate the human mesenchymal stem cells. And he gave them the name human mesenchymal stem cell to correlate with the hematopoietic stem cells that were also described just prior to this time period. The MSCs are also called mesenchymal progenitor cells, multipotential stromal cells, mesenchymal stromal cells, which I think is a little redundant, and then also skeletal stem cells and multipotent adult progenitor cells, or MAPCs. So you'll see these names in the literatures, but they really describe the same group of cells with subtle differences. If you take a bone marrow sample and try to isolate mesenchymal stem cells from them, this is some typical data that young individuals have a lot of these cells in their bone marrow. And after puberty, it goes down quite a bit. I'm out here, so I'm hoping I don't need too many more of mine. But what we found was that these cells have the same potential as the cells here. So there are niches in the bone marrow where these cells can be maintained for long periods of time. And that's also interesting because your whole skeleton turns over about every 6 or 8 years. Most of the tissue in your body turns over. So when you think about these stem cell niches in both for hematopoietic or the mesenchymal stem cells, that those niches are always turned over. And so this is certainly a big opportunity for losing many of those cells, yet some survive. Okay, so when we played out the bone marrow, these small refractile bodies are actually the RBCs, the red cells, and these spindle-shaped cells are the first mesenchymal stem cells that sit down. And eventually you get this lawn of cells, and they're very contact inhibited. So we don't see multi-layered growth the way you might see for many cancerous cells. If you test these by flow cytometry, this population improves and gets greater expression. These are 2 markers we use, CD73 and CD105. 105. But these, what you'll notice is that this population is, you don't see other discrete populations, so it really is a fairly homogeneous population. And yet, for all these flow cytometry data, you'll know that across one of these peaks or one of these populations, you have a 10 or 20 or 30-fold difference in the level of gene expression for most genes that are found on cell surfaces. And that's also shown here. So the cells are typically negative for CD45, 14, and 34, positive for these markers. So these are pretty typical. It's actually beta integrin, and this is very abundant because it's used as the homolog for many of the different alpha integrins. So this is a fairly abundant one. The cells are positive for class 1 molecules, but they're negative for class 2. Yet if you treat the cells with interferon, they do express class 2, and they do express RNA for class 2 when they're not induced. Okay, so they also express telomerase. This is passage 12 cells. These are passage 1 from different donors. And so you can see these ladders of these TTA, GGG repeats. So the telomerase adds these to the ends of the chromosomes during replication. So these are maintained in the later passage MSCs. And this is from a more recent paper. So these are many of the surface molecules that are found on the cells. They are positive for these stage-specific embryonic antigens 3 and 4, but we don't see stage-specific antigen 1 on there. The MSCs produce many factors that we know about, a lot of these interleukins, so they can communicate with many other cells both locally and at greater distance. The colony-stimulating factors, macrophage colony-stimulating factor, granulocyte, and the GM-CSF, these are all produced, and these cells actually are very good at forming a stromal layer for hematopoietic stem cells. So other cytokines that they produce are leukemia inhibitory factor, stem cell factor, FLT3 ligand, TPO, and SDF1. So they also, if you do a genomic screen, they express low levels of proteins found in many differentiated cell types, including cartilage, adipose tissue, bone, endothelial, hematopoiesis support cells, neural cells, and also muscle. So these cells have an open configuration of their DNA, and they're expressing many of these different RNAs at low levels. And if you put them in the right conditions, they can differentiate accordingly. In this lineage diagram, I'd like you to think of these arrows as changes in gene expression. So as these cells begin to differentiate into different cell types, they change their gene expression. When they get down to fully differentiated phenotypes, we've looked for, every gene we've looked for that would be representative of these tissues is represented. So histologically, when it looks like your tissue of interest, you can be sure that it expresses the large majority of the proteins you would expect from those lineages. And these, what you don't see are combinations. So if you were to do something like this with embryonic stem cells, you would get multiple loci that have these differentiation in very local populations of cells. So we don't see, osteogenic cells present in the cartilage lineage. Likewise, we don't see the adipose present in the cartilage lineage. So this comes from a lot of work that these cells produce a lot of growth factors and cytokines, but they also respond to these. And for chondrogenic differentiation, we add TGF-beta. They also are responsive to the basal nutrients that are in the serum, and such as the serum and the glucose levels. We get greater adipogenic differentiation in high glucose. We do not get any differentiation to the chondrocyte lineage if there's serum present. And that's interesting if you think about the fact that cartilage really has no blood vessels in it. So you don't see much serum in your cartilage. And so the cells there really don't see it either. The cells do respond to mechanical forces such as tensile compression and also repetitive cycles of both compression and tension. Cell density is important. The adipogenic differentiation does not occur when the cells are sparse, only when they're confluent. Likewise, the chondrocyte differentiation works best when the cells are in a pellet culture format. These are just saying that we can look at different donors. This is 5 different donors, undifferentiated or differentiated. And each of the donors responds with fidelity and gives us good differentiation as shown over here. So from different donors, we see these differentiations. There's some variation, but when the assays are working well and the technicians are doing everything they should, these assays do work well. This is an example of chondrogenic differentiation, and when it's working well, we get this pearlescent chunk of cartilage. If you take it between your hands and try to crush it, you cannot. If you stick it between 2 pieces of glass, however, you can crush it. And the reason for that is that this is young cartilage. It's not very well cross-linked. So the extracellular matrix is not cross-linked at this point. The thing to notice here is that in high glucose and with TGF-beta, we get large amounts of extracellular matrix produced. If we don't have any TGF-beta, this is what occurs, and really there's no differentiation. In low glucose, low TGF-beta, we have these smaller pellets. So the DNA content in these pellets is the same. It's really just the amount of extracellular matrix that's produced when the conditions are right. So you can think of these cells as occupying a space and waiting for signals to come their way where they might differentiate. And by pushing them in one direction or the other, the cells will respond appropriately. So this is just a visual of trying to graphically show what we've seen in some of the experiments. Okay, so this brings me back to this important point that what we're seeing is where biology is selective and it's not directive. So when you anticipate you want to see something and you don't see it, it's probably because you don't have the conditions correct, and now you've got to select for one process or another that will give you greater differentiation of the cells. So the MSCs suppress mixed lymphocyte reaction and inflammation in vivo. the MSCs are not acutely rejected. So I'll give you some data on that. In a mixed lymphocyte reaction with two different donors, you get a strong T-cell proliferative response. However, if you use MSCs from different donors, you don't see this response from the responding T-cells. So the cells actually can suppress this mixed lymphocyte reaction. And if the cells are differentiated, we worried that, well, they might be rejected at that point. But no, the osteodifferentiated cells still are immunosuppressive. And even if you transduce those cells with the cofactors B7-1 or B7-2, they still will subdue a mixed lymphocyte reaction. We've seen this from many different species, both from rat, canine, goat, baboon, porcine, and human. Mouse MSCs are a little different, so this is a complication, but mouse MSCs are usually not pure. They have some hematopoietic stem cells that co-purify with the MSCs, and it's very difficult to isolate them away. When you do isolate the mesenchymal cells away from the hematopoietic cells, the mesenchymal cells don't grow. So my friends who work on neural regeneration in mice, they can grow up 400,000 cells, and they can do 8 mice, each receiving 50,000 cells. But they can't expand the purified MSCs from mouse. I think this is true for a lot of other mouse studies too, that what I'll say about the MSCs is anything we've seen in these different species, we've also seen in human. So we can't really take our cues from what's occurring with mouse MSCs. Probably you can't read this past the third row, but I'll tell you that we've looked at the engraftment of these cells in baboon, rat, canine, pig, and this is a knockout mouse receiving human cells. But we can detect the cells at 1 month, 3 months, 4 months, 2 months, 6 months, so out to a year. So the cells actually persist in the allo setting. And that's one of the reasons that many of the studies done with MSCs have been done in an allo fashion. I think this is something of a problem. I think a lot more progress could have been made if autologous cells were used in many cases. However, this is difficult with many of these species, and just as it's difficult with man, the cells just aren't ready when you want to transplant. But they're not entirely immunoprivileged. So these are fibroblast cells into bulb C mouse, and these are MSCs into bulb C mouse. And if you look at 10 days, 20 days, and 40 days, you can see the MSCs do persist, but eventually they are lost. The fibroblasts are lost very quickly, so there is a period of privilege for these cells, but it's not, in all cases, it doesn't persist for long periods. Okay. So when my postdoc came to the lab, I wanted her to isolate these different types of T cells or immune responding cells and then test which population the MSCs were really affecting. And so Sudipta was able to isolate T helper 1s, T helper 2s, DC 1s, DC 2s, natural killer cells, and also B cells. This is an example of some of the data, but I bring this up because PGE2, which is an immunosuppressing factor, the MSCs do make some of it, but once you've mixed them with another cell type, there's actually much more produced. And we believe this is actually coming from the PBMCs because we've done this with rats versus human MSCs, rat PBMCs, and with using a rat-specific antibody can detect that there's more of this produced. Likewise, if you take MSCs, they do produce VEGF, but when you mix them with another cell type, you now get very large increase in the level of VEGF. So you can imagine when you have these cells in the lab and you're doing one thing, they are doing one thing, but now when you put them in vivo, now you have an entirely different response that you need to consider. Through Sudipta's work and the work of many others, we now know that the MSCs actually make at least 11 different factors that can suppress the immune response. There's also this peptide LL37 that the MSCs make, and this is an antibacterial peptide. So when patients receive these cells, we don't see increases in bacterial infections, et cetera. This may be part of the story here. The other thing I'll say is that when you separate the T cells from the MSCs, these T cells continue to respond. So you can now challenge them and they will respond appropriately. So it's as if the cells become quiescent when they're in the presence of the MSCs. But when the MSCs are removed, the T cells are now able to respond. So there's an increase in the Tregs as well as the Th2 cells, and this is also responsible for some of the suppression of the immune response that we see. Now, this is a graft versus host experiment. It's actually a patient study published by Katerina LeBlanc in Lancet. This was a 9-year-old boy who had leukemia and was transplanted with third-party hematopoietic population. The child developed graft versus host disease between 30 and 60 days, and they were given MSCs from the mother. So essentially this was 60 million cells in a 30-kilogram child. The cells were from the mother, and the child responded by quickly suppressing the graft versus host disease. And so this was, again, a child that had grade 4 GVHD would be expected to pass probably within 3 or 4 days. At some point, they removed the cyclosporin to try to get a greater graft versus leukemia effect. The child then responded with graft versus host disease, whereupon he was retreated with the remaining number of cells. Here it was 30 million. Again, you see the suppression of this graft versus host disease. And this child then went home, better part of 2/3 of a year, but was able to return home. So since that time, there's been a number of different studies clinically to look at graft versus host disease clinical studies. Multiple infusion of MSCs induced sustained remission in children with steroid refractory grade 4 graft versus host disease. There was also a phase 2 study done with by the European group, and there was also a commercial MSC product produced by Osiris Therapeutics, which didn't meet its endpoints. So it was approved in Canada, Switzerland, New Zealand, and Korea, but not all of its endpoints. It's now only used for compassionate use in the US. Osiris no longer produces this. It's now licensed to another stem cell company called Mesoblast, which is in the US, but also an Australian company. Okay, so one of the interesting things about the MSC is that they actually seem to migrate to sites of injury. It may be that they don't migrate so much as they find a place and they attach there, and then it's hard to remove them. It could also be that when you have tissue damage, a lot of the capillaries become leaky, and when the cells get there, they stay there because it's very hard for them to crawl back in against any pressure gradient. But they do accumulate at sites of injury. And they, as I've shown you, they suppress inflammation, and so that's probably a good portion of what they do. And in doing so, they can actually improve tissue repair and function. So I've been working in the heart for a number of years. We have different ways to, the beta blockers and ACE inhibitors work very well. Reperfusion is always important. If we have to get to surgery, we can go through coronary artery bypass. There are implantable pumps that can be used, but cell therapy is really a new thing to the armamentarium of the cardiologist. You can use skeletal myoblasts, and this has been tried. This led to arrhythmias because the skeletal myoblasts do not respond. They don't recover the same way that cardiac cells do. So they can't be used long-term in the heart. Neonatal cardiomyocytes are difficult to come by. Different types of stem cells have been used. And I'll show you some data on mesenchymal stem cells, both in rat, pig, and the sheep infarct model. Of the 180 ongoing clinical trials with MSCs, about 17 of them are for the heart, and the University of Maryland Medical Center is participating in 2 of these. So this is a model, the drawing of the heart. When we tie off this left anterior descending artery, this region of the heart is at risk, and you get an infarct here, and many of these cardiomyocytes are dying. In general, these infarcts are carried out for about an hour in the rats and the pigs, and then you allow reperfusion. So much as we would expect to see in the clinical setting, reperfusion would be the first event that would occur for the patients. If we take MSCs that are allogeneic, that is ACI rat MSCs, which are labeled with LAC-Z, which is the blue dye here, and we infuse those into Fisher rats by a tail vein, we can then take a bone marrow sample at, say, 10 days or somewhere in this range, and we can find those blue cells that are found in the bone marrow at this point. If we then do the cardiac infarct in these animals and then come back and look 2 weeks later, so again, we've tied off here and then allowed reperfusion. And if we stain the whole heart, what you see is all those blue cells are found where the infarct was. All right, so, and if you section through this, you can see it's a full thickness defect and that the cells are throughout in this region. Oh, here it is. These were dye-labeled cells. That's what the red is. Phospholambam is what the green is, which is a calcium handling protein in the heart. So what you find is that the MSCs are found in the infarcts. They're found where there's space for them to be. They're not found in the healthy regions of the cell. We see hints of expression of cardiac proteins, and I'll show you a little bit more of this. But what we don't see is striations or new cardiomyocytes being formed in these regions. Okay, and the pig model is larger. We can actually put in instrumentation and get piezoelectric crystals and now get measurements across the wall in the infarct region. And when you do these infarcts, all this tissue is at risk. You begin to see collagen accumulation and you see thinning of the wall. You'll see some compensatory compensation from the other side of the wall. This hypertrophy is allowing greater pumping action than would be if you only had this starting amount of muscle. But it's pushing against this wall that does not move, and it's trying to expel all the blood from the heart. And so here's, in both of these, to orient you, this is about midway across the heart. It's one of those sections that we were looking at. 12 o'clock would be about here, and this is where the infarcts are in both animals. Let me start this one. And you see there's a regular pumping action, but this side is twitching, but it's not contracting the way that this side of the heart is. These animals that were treated with MSCs, you can see that it's a much more dynamic and circular contraction. You can see the infarct is still present, but the cells that survived in this region are still contractile. This is a much more efficient pump. The data looks much better, and the echo technician was blinded to these animals, but each time she could tell which ones received the cells and which did not. Okay, so taking some measurements afterwards over a period of time, this is 4 weeks, or as early as 4 weeks. We've gone longer. But what we see is that the cells, the animals that receive the heart, the hearts that receive the MSCs show greater, in this case, thickness in the region during end-diastolic wall thickening. Those animals that did not get the cells continue to get weaker and weaker, and that's due to the dilation. And this is this wall thickening that we talked about at base. Again, the untreated animals get worse. Those treated animals stay about the same or maybe even improve a little bit. We've done this in sheep model more recently, and here we're looking at the strain that occurs. So this reduction in strain is a good thing. Here, those animals that did not get the cells, again, the strain increases, and that can be detrimental to the pumping of the heart and deleterious remodeling. I'm not going to go through this in detail, but just to say that if we look at hypertrophy-related molecules or calcium-handling molecules and apoptosis of the cardiomyocytes, that in all cases that the animals that received the MSCs fared better than those animals that did not receive the MSCs. This again was allo cells going into the sheep model. Okay, so what's missing? Well, we can detect, in this case, myosin heavy chain in red, alpha-actinin, tropomyosin, phospholamban, as I showed you earlier. So all of these are molecules that are structurally important for sarcomeric organization. We don't see sarcomeres being formed at this time frame. And so I guess there's something that's still missing, but this is an adult animal. It's not an early embryonic animal, or let's say someone who was a very young animal that might still modify its production of new sarcomeres. So there may be a factor that we're missing. It could be an external one, something that's involved in registering the sarcomeres with different animals and allowing those to organize. And those are known in the field, but we really haven't looked at those. Okay, so this is a little different. This is a homing experiment in a dog model. So it's a 90-minute LAD occlusion, and this is actually by a balloon catheter. So this is a little different than the other model, which was an open animal. They've been infused with 150 million cells that were labeled with oxine and also Feradex, and this allows us to image them either by MRI or by this SPECT imaging. And this gives you spectacular images. So the cells are first found here in the lungs, and then as they're infused, and then they work their way into the heart, as you'll see here. So as early as one day, there's a little bit accumulating in the heart, but really at day 2, you can really see these cells. And later time points, a little longer exposure, but you can see the cells are there in the heart. So that's all good. Here's the bad news, and that is that only a very small amount of the cells remain in the infarcted hearts. And we've lost the majority of the cells, and that's what you're actually looking at here. So the question is whether this is all the cells that can be retained at that site, or looking at it another way, would this mean we only need to infuse this many cells to get the same therapeutic benefit? And that's something we really don't know. Is this large bolus of cells pushing cells, the MSCs, into the tissue where they're needed, or would they go there automatically? I don't know the answer. Okay, so infusion engraftment remains a problem. The MSCs have a strong safety profile in clinical trials. Let's take a quick look at that. As I mentioned, skeletal myocytes do not give you very good results. Those have all been abandoned. Cell type, timing, and the dosing are all important for these different models that you might want to use. The published studies have been from a variety of groups, but Josh Hare was involved. He was at Hopkins when he started these studies, but he's now at Miami. So this was an open heart surgery similar to the pig model that I showed you. These are actually the catheter-delivered study that's now come out. And these are all phase 1s, but now there's a phase 2 study ongoing. Different types of cells are actually being used depending on who's sponsoring the trial. In all cases, we've seen small but significant improvements in the heart output and heart function. These are not large enough, but given the small number of cells that actually are retained there, it's actually pretty good. So if we can improve the engraftment, I think we can move these numbers way up. This was a meta-study that looked at 36 different studies. The only thing that they said was that was common was transient fever following the infusion of the cells. But really, they too, I think, were impressed by the fact that they didn't see more detrimental effects of these cell infusions. So I think where we're at is now developing improvements for second-generation cell therapies with the MSCs. And I think that's, yeah, so just again, to go over a few of these things that they're more rare than MSCs. You can expand the populations in vitro. They're found in all tissues, and they produce lots of growth factor cytokines and anti-inflammatories. The infusion of these cells results in great cell loss, especially in vitro, if in vitro expanded. So when we think about MSCs or HSCs, you might also think about the CAR T cells that are now winding their ways through therapies. So I think when we culture cells in vitro and now we're trying to use them in vivo, really need to look carefully at the best way to get these cells that we've worked so hard to produce to do what they wanted, what we'd like them to do in vivo. And I think that's where many of the payoffs will come in the next couple of years. I'll mention also that the adipose-derived stromal vascular fraction, again, this fraction that's evolved from the isolation of these vascular fractions, so you've gotten rid of all the fat cells, this may provide enough autologous cells without the need for in vitro expansion. Okay, and I'd like to thank my collaborators at the University of Maryland, also at Johns Hopkins, University of Florida, Pittsburgh, and also those people who were at Osiris at the time. I think that's it. Thank you. Any questions? No questions? All right. Thank you.
Oh, that's a nice picture.